Good morning. I'm John. I'm going to present work this morning from my lab at NYU and in collaboration with Kendrick Kay. Okay, I'm going to begin with visual perception, which is where I began as a student. Perception, seeing, is an interpretation. It requires combining image data over space. This is a visual illusion that reflects combining information over space. The upper pieces may look white to you, the lower pieces may look black. If you zoom in and add an outline to prevent your visual system from combining over space, you see the illusion goes away and the pixel values are in fact the same. Similarly, seeing depends on combining information over time. The display on the left probably looks like one dot moving back and forth. The one on the right looks like two flashing dots. They're the same spatial pattern, but they just differ in the temporal pattern. One is in synchrony, and one is out of phase, one is in phase. And we presume that these computations, analyzing the image data, combining over space and time, depends on computations in cortical circuits distributed over many maps. And as we know, and as we've seen throughout this conference, there's been incredible progress in making measurements from the living human brain, from the days of PET imaging in the 1980s with a effective resolution of approximately a centimeter and a half to modern neuroimaging at high field, say, where we can measure the whole brain at less than 2 millimeters with 1 second sampling and get multiple retinotopic maps out of a single session. But just as important as the advances in resolution and acquisition are the advances in software and modeling. One example is the population receptive field. This is a model that relates the input, the stimulus, to the observed fMRI time series through a spatial model, here a 2D Gaussian parameterized by x, y, and a size sigma. You can see this is a poor model, but if we change the position, it does better, and if we change the size, it does better still. And with software and about a few hours of analysis, you can solve for these parameters in thousands of voxels from a subject. If you aggregate those PRF model solutions, you find nice systematic patterns. For example, as the eccentricity increases from the fovea here on the x-axis out toward the periphery, the receptive field size increases here on the y-axis, and it also increases as you move through the visual system from, say, V1 to V2 in extrastriate maps. Now, if this is all we had learned, you might have said, okay, that's nice, but didn't we kind of know that from 50 years of neuroscience? And I would say yes, but you know what? Since the method was introduced, we've made a lot of progress and have started to use it to answer a lot of new questions, which I have co-authored a couple of reviews about, where we've described new advances in map discovery, in parietal, frontal cortex, even in the last month, 5 visual maps in the cerebellum, where receptive field models have been used to analyze faces, scenes, and word-responsive regions in the temporal lobe. We've used it to study task, disease, development and aging, plasticity and sight restoration, many topics where we actually did not know the answer 20 years ago from animal literature. So once the method gets established, it becomes very useful to apply to new domains. Now, as great as that sounds, I'm going to move immediately to a model failure, because we learn a lot from the model failures. And this example of a failure is a failure of linearity. And I'll illustrate the assumption here. If you have a V1 voxel and measure its response to a stimulus part, and then measure its response to another stimulus part, the PRF model I showed you implicitly assumes that the response to the full stimulus would be the sum of those responses. But it's not. The measured response is less, about 20% less in V1, and an even larger failure of linearity as you move through the visual system. Okay? These failures can be captured by a slight change to the model, where we take our pointwise multiplication of the stimulus in PRF and add an output nonlinearity. And I'll illustrate the idea of that. If you have a small stimulus and a large stimulus, the linear prediction would be about that the response should double. But once you put it through the nonlinearity onto the y-axis, the responses are much more similar, and that is accurately reflected in the data and can, in fact, generalize to many new stimuli. So what we did was we took models fit to simple stimuli and then cross-validated them on stimuli that varied in size from small to large like these, and show the pooled response in several visual field maps. When we use the linear model, it does okay in V1, but pretty bad in V4 and quite bad in MT. The CSS, the compressive spatial summation model, does very well in all areas and shows something like a tolerance to changes in size and position as you move through later areas of the visual system. So we can make a cartoon of that and show that as you go from early to later visual areas, the spatial pooling gets larger, the compressive nonlinearity becomes sharper, and that's consistent with increasing tolerance to changes in position and size. Okay, with that under our belt, let's turn to time. And we're going to use the same methods to study temporal processing using fMRI. And like Ben said, even though the measurement doesn't have fine temporal resolution at the millisecond scale, the amplitude is sensitive to millisecond changes in the stimulus timing. And we varied our stimuli from one video frame, 17 milliseconds, up to about half a second, or two stimulus pulses with variable interstimulus interval, and measured the bold response in different visual areas. We can do the same test of linearity, and when we do that, we see that in V1, the response to a stimulus that doubles in length doesn't double in amplitude, it's less than double. The linear prediction fails. And in higher visual areas like temporal occipital cortex, the linear prediction fails even more. There's not much increase as the stimulus gets longer. And we can capture that with an analogous model. We had the compressive spatial summation, and for this we explain the data with a compressive temporal summation model. Instead of a 2D stimulus aperture, the input is a 1D time course, and instead of a 2D PRF, the model is a 1D impulse response function, and then it goes through a nonlinearity. We use divisive normalization instead of a power law. You can ask me about that at another time. It behaves very similarly. And the model actually works quite well. I don't have time to get into this, but I want to contrast this with another model developed by Hiroshi Horiguchi on luminance. Kalanit, who will talk about this in more detail in the next talk, on contrast patterns, which is a two temporal channels model, which I think also has a lot of merit. Okay, in our model, we find that as the stimulus duration increases, the linear model underpredicts the response to short stimuli and overpredicts the response to long stimuli, but the compressive temporal summation gets it quite accurately. As we look at the stimuli, the two-pulse stimuli, we also see the CTS model is more accurate. The linear model is flat because the total duration is always the same. And you notice something interesting, which is that as simple as this model is, it captures an interesting phenomenon whereby short pulses with a short ISI have a reduced response like adaptation, and a longer ISI, you see recovery from adaptation, okay, implemented just with a simple output nonlinearity. And the model fits well not just in V1, but across visual field maps. And we can see that just as in space, the case of space, the difference between the linear model and the compressive model is modest in early visual areas like V1, and it gets increasingly large down here on the bottom. You can see in TO, temporal occipital, and IPS, the difference between the models is quite large. Okay, and we can summarize that by showing that as you go from early visual areas to later visual areas, the models become, and the data, more nonlinear, more adaptation, and the temporal window length increases from short to longer. Okay, so we can show that with a cartoon where early visual areas integrate over small regions of space, but also relatively short regions of time with a weak compressive nonlinearity. And in later visual areas, the information is combined over larger regions of space and time with a stronger compressive nonlinearity. And we speculate that based on this similar pattern for space and for time, Cortex is using a similar processing strategy to achieve higher level and increasingly invariant representations of the visual world. Now, those measurements from fMRI were sensitive to slight changes in stimulus duration and ISI, but they didn't reveal the millisecond timescale of the responses. Okay, that was implicit in the model, but we couldn't test it. And when we actually visualize those implicit responses here on the left, it turns out not to look that much like the data. This is ECOG broadband data from a V1 electrode. And we're surprised. On the one hand, the model very accurately predicted the fMRI responses. On the other hand, it doesn't look that much like the time-resolved data. And what we inferred was that the fMRI model is correctly getting the amount of adaptation right, but what it's not getting right is the exact timing of when the adaptation kicks in. So following this theme of making slight additions to our model, we took our compressive temporal model here on top and added one element, a low-pass filter, which just delays the time at which the inhibition kicks in. And as a result of that, you get a different kind of predicted output. The early response reflects the linear filter summing, and then once the normalization or nonlinearity kicks in, the response goes down. And that matches the ECOG data pretty well. You can see here that they look very similar, but in red is the model, and in black is the data, and that's an average of 12 V1 electrodes from about 3 different ECOG subjects. So that was V1. It also does a good job in later visual areas, V2, V3, and anterior maps. You can see that on the top row, we aggregated the anterior maps because we didn't have many channels from each separate map. And on the bottom, if you plot them together, you can see slight increases in the timing from V1 to V3, and then a larger increase in the anterior maps reflected down here in the middle plot on the bottom quantitatively. And in agreement with the fMRI data, you can also see a greater extent of adaptation in the later visual areas compared to the early ones. Now, remember I said earlier I like to point out model failures. You may notice here our stimulus was on for half a second. There's a little bump when the stimulus goes off. And that makes me think of Hiroshi and Kalaniit because their 2 temporal channels model captures that offset response. And we've discussed that, and we think we need to integrate that aspect of the model with our delayed normalization model to accurately account for all the data. Okay, so I just want to conclude with a couple of thoughts. One is that we use this normalization model. And I think it's important that the model we formed is relatively simple and interpretable, different from a deep neural network model, for example. Not that there's anything wrong with that. And this normalization model discovered or put forth in the early 90s from David Heeger has been since used in many sensory modalities, vision, hearing, smell, and also cognitive domains. So it has a lot of legs, and we think it's a general cortical processing strategy. Moreover, the delayed normalization has also been used as a model for other parts of cortical processing, including some work from Paul Glimcher's lab, where they looked at monkey spiking data and show as a function of reward, so it's milliliters of juice reward. There's an initial transient, and then the response goes down. And that looks kind of like the visual ECOG data I showed you, and they model that with a delayed, or they call it a dynamic normalization model, which shows the response increasing due to linear summation and then decreasing as the normalization kicks in. So we think that this strategy of processing is probably widespread in the cortex, and we think it's therefore important that we can make connections between visual processing and other cortical processing. Now, as I pointed out earlier, in the spatial domain, we first developed these PRF models. There was a certain amount of pushback from certain people. Well, what have we really learned that's new, right? And then over the next decade, there was an explosion of findings with interesting new results that weren't known before. I think in the temporal domain, we're several years behind. There has been important work. I'm looking at Uri Hassan here in the front, who's done important work on hierarchical processing of temporal in the temporal domain. But I think it's going to be over the next 5 to 10 years that we see some of these same developments in the temporal domain, where the models, for example, are applied to disease, to retinal and cortical lesions, optic neuritis, psychiatric conditions. Our lab is working on integrating these models with spatial processing to understand the dynamics of recognition. And we're also hoping to develop more normative theory that can explain why it's useful to have a response like this initial transient followed by a slower sustained response, and why it's useful to have this hierarchy of processing scales. And finally, I want to end by pointing to Iris Groom, sitting here in front. She will be giving a talk on new work that follows in this tradition, but has much richer ECOG data. She's giving a talk in this morning's session, Mapping Sensation, Perception, and Attention, and also has a poster this afternoon. And with that, I'd like to thank my funders and you for coming this morning. Thank you.